Chapter 12, Section 4, Meiosis. Learning objectives. Identify how many sets of genes are found in most adult organisms. Describe the events that occur during each phase of meiosis. Identify the differences between meiosis and mitosis. Explain two alleles different genes can be inherited together. Each cell from the fruit fly Drosophila has 8 chromosomes. 4 of these chromosomes came from its male parent, and 4 came from its female parent. These two sets of chromosomes are homologous, meaning that each of the 4 chromosomes from the male parent has a corresponding chromosome from the female parent. A cell from 2 sets of homologous chromosomes is said to be diploid, meaning double. The diploid cells of most adult organisms contain 2 complete sets of inherited chromosomes and 2 complete sets of genes. The diploid number of chromosomes is sometimes represented by the symbol 2n. Thus, for Drosophila, the diploid number is 8, or 2n equals 8, where n represents the single set of chromosomes found in a sperm or egg cell. Some cells have a single set of chromosomes, and therefore a single set of genes. Such cells are haploid, meaning single. The gametes of sexually reproducing organisms, including fruit flies and peas, are haploid. Remember that mitosis has 4 main phases: prophase, metaphase, anaphase, and telophase. Starts with interphase. An interphase is responsible for the growing of the cell, the duplicating of the chromosomes, and it's also responsible for having 2 daughter cells that are genetically identical for the original cell. How do sexually reproducing organisms produce haploid gamete cells from diploid cells? That's where meiosis comes in. Meiosis is the process in which the number of chromosomes per cell is cut in half through the separation of homologous chromosomes in a diploid cell. Meiosis usually involves 2 distinct divisions called meiosis I and meiosis II. By the end of meiosis, a single diploid cell has produced 4 haploid cells. Just prior to meiosis I, the cell undergoes a round of chromosome replication during interphase. Each replicated chromosome from the male and the female consists of 2 identical chromatids joined at the center. After interphase I, the cell begins to divide, and the chromosomes pair up. In prophase I of meiosis, each replicated chromosome pairs with its corresponding homologous chromosome. This pairing forms a structure called a tetrad, which contains 4 chromatids. As the chromosomes pair, they sometimes undergo a process called crossing over, in which bits and pieces of homologous chromosomes are exchanged. Crossing over produces new combinations of alleles on each chromosome. Crossing over is the process in which causes you to look different from your parents. As prophase I ends, a spindle forms and attaches to each tetrad. During metaphase I of meiosis, paired homologous chromosomes line up across the center of the cell. As the cell moves into anaphase I, the homologous pairs of chromosomes separate. During anaphase I, spindle fibers pull each homologous chromosome pair towards opposite ends of the cell. When anaphase I is complete, the separated chromosomes cluster at opposite ends of the cell. The next phase is telophase I, in which a nuclear membrane forms around each cluster of chromosomes. Cytokinesis follows, forming 2 new cells. Meiosis I produces 2 daughter cells. However, because each pair of homologous chromosomes was separated, neither cell has the 2 complete sets of chromosomes in a diploid cell. Those 2 sets have been shuffled and sorted almost like a deck of cards. the two cells produced by meiosis I have sets of chromosomes and alleles that differ from each other and form the diploid cell that entered meiosis I. The two cells now enter a second mitotic division called meiosis II, which is shown in this picture. Unlike the first division, neither cell goes through a round of chromosome replication before entering meiosis II. As a cell enters prophase II, their chromosomes each consist of two chromatids becoming visible. The chromosomes do not pair to form tetrads, because the homologous pair were already separated during meiosis I. During metaphase of meiosis II, chromosomes light up at the center of each cell. As the cell enters anaphase, the paired chromatids separate. The final 4 phases of meiosis II are similar to those of meiosis I. However, the result is 4 haploid daughter cells. Each of the 4 daughter cells produced in meiosis II receives 2 chromosomes. These 4 daughter cells now contain the haploid number, or N, just 2 chromosomes each. The haploid cells produced by meiosis develop into the gametes for sexual reproduction. In males, these gametes are usually called sperm. In females, these are known as egg cells. The words mitosis and meiosis may sound similar, but the two processes are very different. Mitosis can be a form of asexual reproduction, whereas meiosis is an early step in sexual reproduction. Mitosis and meiosis also differ in the way chromosomes move and in their number of cell divisions. A cell replicates or copies all of its chromosomes before entering either mitosis or meiosis. However, the next step differs dramatically. In mitosis, each daughter cell receives a complete diploid set of chromosomes. In meiosis, homologous chromosomes are separated, and each daughter cell receives only a haploid set of chromosomes. As a result, in meiosis, the 2 alleles for each gene are segregated and end up in different gamete cells. The sorting and recombination of genes that takes place in meiosis helps to increase genetic variation from one generation to the next. Mitosis does not change the chromosome number of the original cell. Meiosis reduces the chromosome number by half. Mitosis results in the production of 2 genetically identical diploid cells, whereas meiosis produces 4 genetically different haploid cells. A man by the name of Thomas Hunt Morgan first realized in 1910 that the Drosophila, a type of fruit fly, had a thing called gene linkage. Morgan identified more than 50 genes and the traits that they caused. He discovered that many of the genes appeared to be linked together in ways that, at first glance, seemed to violate the principle of independent assortment. Morgan's findings led to 2 remarkable conclusions. First, each chromosome is actually a group of linked genes. Second, Mendel's principle of independent assortment still holds true. It is the chromosome, however, that is sorted independently, not the individual genes. Alleles of different genes tend to be linked together from one generation to the next when those genes are located on the same chromosome. In 1911, a Columbia University student was working part-time in Morgan's lab. This student, Alfred Sturtevant, wondered if the frequency of crossing over between genes during meiosis might be a clue to the gene's location. Sturtevant reasoned that the farther apart two genes were on a chromosome, the more likely it would be that crossing over would occur between them. If two genes are close together, then crossing over between them should be rare. If two genes are far apart, then crossing over between them should be more common, reducing the frequency in which they are linked. By this reasoning, he could use the frequency of crossing over between genes to determine their distance from each other. Sturtevant gathered up several notebooks of lab data and took them back to his room. The next morning, he presented Morgan with a gene map showing the relative locations of each known gene for one of the Drosophila chromosomes. Sturtevant's method has been used today to construct gene maps. A gene map shows the location of a variety of genes on a chromosome.